Hey guys, this is your review that you're doing with Miss Small. I'm going to go over the answers. That's going to really help you on your 4th 6 weeks common assessment. So starting with vocabulary, I'm going to go over these ones pretty quick because I think we know most of these in here. So heterozygous, you have to remember the prefix hetero means different. So that's when you're looking at a genotype and there are 2 different alleles. That is letter D. Homozygous, we got to remember homo means the same. So that's when you have 2 same alleles, either 2 dominant or 2 recessive. Looks like letter B. Now an allele is a form of a gene. For example, let me draw off to the side here in blue. If we had a gene for, I don't know, eye color, the alleles are forms of that gene, such as blue and brown. So different forms of a gene. It's letter F, mitosis. We know toe is a good way to remember that. Toe is part of our body, so mitosis makes body cells. Key here is that all of our body cells are identical. Going on to meiosis. Meiosis must be the opposite, which is making sex cells. And that looks like that is letter C. If you don't remember gametes, gametes are a scientific word for sex cells, and those are all genetically different. Genetic recombination, guys, that is the same thing as crossing over or making new combinations of DNA. Homologous structure. Now, that is talking about evolution. Again, we know homo means same or similar. That's how we tell things have a common ancestor. Is they have a similar structure. They have a different function, but that doesn't matter because remember structure. If we can tell structure, then we can also tell, I'm going to draw an arrow, that they have similar traits. A structure is a trait. That means they have similar DNA. And we know things with similar DNA are closely related. Haploid, well, hap means half. So gametes have half the number of chromosomes. That's why they're called haploid sex cells. That's letter J. Evolution is the process of genetic change over time. That's I. And a chromosome. Now a chromosome, if I were to take DNA, I'm going to draw over here. DNA looks something like that. I wind it up super, super tightly because DNA would take up many, many football fields if I stretched it out. When I wind it up tightly, it can look like this or it can look like that. That is a chromosome. All it is wound up DNA. So that right there is our vocabulary review. Now going on to the tic-tac-toe, I'm going to go over all the questions. I know you didn't have to do them all, but let's just go ahead and go over them. So the group that is structured, well, this is DNA. How do I know that? Is because it is double-stranded. There are 3 parts of DNA, okay? A phosphate, a sugar, which is our deoxyribose, and a nitrogenous base, which those are the letters A, T, G, and C. So let me just draw some arrows to each of these. So the hydrogen bonds are actually these right here. They connect the 2 nitrogenous bases. Now the phosphates, let me go ahead and color the phosphates in red. The phosphates are these little circles right here. Usually they're going to have a P on them to signal a phosphate. Deoxyribose, that's the sugar. Let me go ahead and color those in green. It's going to be right here. Remember, the sugar looks like a hexagon. And lastly, the thing that has a circle around it, I'll draw another circle right here, guys, in the middle. Those are my nitrogenous bases. So the answer here is D. Those stand for the A, the T, the G, and the C. Let's continue over here. Before I go over the answer, let's go over these mutations. Now, a mutation we know is a change in DNA. So one answer choice I'm going to cross out is translation. We know that's when I go from RNA to DNA. That doesn't make sense. Now, if we recall, a point mutation is when I substitute a letter. We know insertion, right? Insert means to add, and delete means to remove. Remember these two right here, guys, those are frame shift. Those are the ones that are more damaging because they shift everything over. So let's go back up to the question. I have normal hemoglobin, DNA, sickle cell, hemoglobin. That's a disease. I have their DNA. So let's go ahead and compare. Right here, it looks like, boom, that letter A is my mutation. As you can see, I change it from a T to an A. I don't add anything. I don't delete anything. I just swap it out. So the correct answer is a point mutation. That is a substitution. Now, let's go over here. A little review between active and passive. So I'm going to draw this hill. Remember, guys, I have my mountain here, the low points here, the high points. I have a ball rolling up the mountain. That is going to take energy, or ATP. So that is our active. And on this side, I have a ball rolling down the mountain. That is no ATP. What doesn't take energy, like sleeping, that is passive. So two differences there. Passive is no energy when you move from low to high concentration. High to low concentration. Active is the opposite, using ATP energy when you move from low to high, going up the mountain. Cancer is a disorder in which some cells have lost their ability to control their... Now, cancer is something, guys, that we didn't talk about. It was in your homework, but you have this cell cycle, right? It goes something like G1, S, G2, mitosis, cytokinesis. And this is the process of making new cells for growth and to replace dead ones. Well, what happens, guys, along that cell cycle, we know checkpoints that check for errors. If there are errors, it's going to stop them and fix them. But some mutations don't get caught. The mutations that cause this cell cycle to go way too fast, like out of control, you make way too many cells way too fast, that mutation gives rise to cancer. So, in cancer, you have lost their ability to control the rate, which means the speed of cell division. It goes way too fast. Now, when you have too many cells that you don't need, those cells are going to clump up just like this. When you have cells clump up like that, that is called a tumor. That's all you need to know right now about cancer. Continuing, a clone plant has a diploid. Remember, di means 2. So, clone plant has a diploid chromosome number of 12. When the diploid chromosome number of the plant cell that was used to produce the clone plant. So here, I have a plant. I know that's not a good plant. There's my plant that I made. And I made that plant by taking another plant and cloning it, or making an exact copy. Well, it told you my new plant has a diploid number of 12. Another way to say that, diploid is body cells. So their body cells have 12 chromosomes. That means the plant that it came from also has 12 chromosomes in its body cells, if they're exact copies of each other. Animal reproduction depends on gametes. So gametes, you should automatically be thinking sex cells. When you think of making sex cells, you're thinking of meiosis. Crossing over. Now let me draw crossing over on the side here. It's literally when two chromosomes, homologous chromosomes, buddy up, and they end up doing an exchange of their genetic information. So it looks something like this. We also call that genetic recombination because you're making new combinations of DNA, getting that variation. So crossing over leads to an increase in genetic variation. We know variation means differences. Genetic is DNA. So we're making differences in DNA. Next, mitotic, so I'm thinking meiosis, cell division results in gametes that produce sperm and egg. So sperm and egg cells are obviously sex cells, and we know a word to describe sex cells are haploid. They have half the amount of chromosomes. So when they combine a sperm and an egg, your zygote will have a complete 2 sets of chromosomes that it needs. One from the mom and one from the dad. So another way to say haploid, or cutting it in half, is we're reducing the number by 50%. Last problem here, which statement best describes inheritance in humans? So this word inheritance means passing down from your parents. So offspring, so it's talking about the offspring and the genes we inherit. Well, we know we have 2 alleles for every gene. For example, my eyes are blue. My genotype could be big B, little b. One of those alleles I got from mom and one I got from dad. So I have 2 alleles for every gene. One is coming from each parent. So let's see what says that. So they don't come from the same parent, right? So we can go ahead and cross off that. And we know this letter A says I'm getting 2 from each parent. Well, then I would have a total of 4, like big B, little b, little b. We know that's not true. I get 1 from each parent. For example, big B is the gene for eye color. 2 alleles, 1 from each parent. All right, guys, let's go on to the back. I'm only going to go over the ones that I think we might struggle on a little. The rest I can go ahead and just tell you the answers to. So let me start off with this one at the top. Now, replication. Replication, we have to remember, guys, is when I go from DNA to DNA. So your answer here should have T's in it, right? It shouldn't have U's because I'm making DNA. Another key, we know everything's opposite. So T goes with A, G's goes with C's. Apples in the tree, car in the garage. You also want to make sure these 3 and 5s are opposite, okay? So the answer here, if we did it correct, we know my answer should have T's in it. And it also needs to go a 3 and a 5. So that looks like letter B. Another one, guys, I want to go over is right down here. In a forest, grasses are autotrophs. Autotrophs is another word for producer. Grasshoppers are herbivores. We know that means you eat plants. Snakes are secondary consumers and hawks are tertiary consumers. Draw a food pyramid to represent these. All right, I'm going to make mine a little bigger over here. All right, so let's go ahead and... All right, so producers are at the bottom, so this is grass. Then above it I have my herbivores. Grasshoppers, and then it goes secondary. The primary ones are the grasshoppers, after snakes. And then we have tertiary at the top, which is going to be the hawks. Perfect. All right, another one. Who's the father? Which of the father's pattern of DNA is closest to babies? So we're not going to go into detail about this, but this is another way to show how closely people are related. And we know the more similar your DNA is, the closer related. So in this device, it's a way to show kind of the different weights of your DNA. And if your DNA, the pattern is very close to someone else's pattern, that means they're related. So let's look at babies, and then we have 3 options of father. So I want to see who's the father. What pattern is closest related to the babies? Well, father 2 doesn't look anything like it. Looking at babies, neither does really father 3. So I'm going to go with father 1 is the father. The pattern, you can see right here, is most closely related to the babies. And the last one that I want us to get set up on is, let's go to this one right here, this cladogram, okay? Which statement is best supported in the information? I'm sorry, it's a dendrogram. So remember, the closer you are, like the gray wolf and the coyote, that means the more you're related. So let's go to letter A. The African wild dogs are more genetically similar to the golden jacks. Okay, let me underline those in blue. The African wild dogs are more similar to the golden jacks than they are to D-holes. Well, we know that's not true. The African wild dog is way closer to the D-hole, so that's not true. Dogs, okay, I'm going to do that in green. The dogs are more genetically similar to the Ethiopian wolves than they are to gray wolves. That's also not true. The dogs are way closer in this picture to the gray wolves than they are to the Ethiopian wolves. Let's go to C. Coyotes. Okay, so I put a mark in yellow. Coyotes are more similar to gray wolves than they are to Ethiopian wolves. Well, let's go back and see when the gray wolves and the coyotes split. And that looks like right about here. And let's see when the gray wolves and the Ethiopian wolves split. That is all the way back here. So remember, the less time ago you split, the more similar you are because you've had less time to change. So that is actually true. And the last one, I'll just show you how to set it up, right? I want to go over this dihybrid cross. We know we're going to have to use a dihybrid because here we have 2 traits, correct? So before you go ahead and go into your dihybrid, which is going to look like this, correct? I want you to make sure you have the 2 parents, T, T, Y, Y, and T, T, Y, Y. That before you fill out your cross, you're filling out the 4 combinations for each parent. I hope this helped. If you have any further questions, please either text me or stop by my classroom, and I'd be happy to help you out.